this webinar series. Today we'll have two presenters. And to begin, we are welcome to stay mute until the two presentations are over. And at the end of the presentations, the Q&A will be opened for questions from the presenters, and further information will be communicated at the end. So I will hand over to Marie-Noel to introduce today's presenters.
Thank you. Thank you, Clinton. Good morning, ladies and gentlemen. Good afternoon, good evening, wherever you are. I'm so happy to see you again this morning. It's a morning for me. So today we will have three presenters, three scientists from ICRISAT who will present on the use of GSI, also to understand the diversity of the collection and the developing subset. So without further delay, I will introduce Dr. Kuldeep, who is a principal scientist at ICRISAT and also a Genebanks manager. Kuldeep, you have the floor. Thank you.
Thank you. Thank you, Marie, and thank you, Abhina. Well, good morning, good afternoon, good evening to you, wherever you are. And I'm here with my ICRISAT team today. My two colleagues, our presentation will be in three parts, 20 minutes each. Me, then my colleagues Damaris and Vetri. They are the nicknames, and of course, both of them are scientists and senior scientists, principal scientists here at ICRISAT. What we'll be doing, talking more about the crops, I mean, all the principles, those will be related to crops. We work at ICRISAT. And the biggest advantage is that we work with the crops which have the different modes of pollination. That means they are both self-pollinated, often cross-pollinated and cross-pollinated. And so you will have an advantage of really learning and asking any sort of question on any of those mechanisms. Let me share my presentation with you. Marie-Noel, is it visible?
Yes, Kuldeep, you can go ahead.
Good.
Yeah.
Okay, so what we'll be talking more on conserving the genetic structure of the collections, and then we'll see what are the applications of the genomics in determining the population sizes which are required for regeneration. So let me take you this slide, which is the essential activities of a gene bank. So we classify them into 8 distinct activities. Acquiring are the collections of the germplasm, conservation of the germplasm, characterization, regeneration, evaluation, distribution, safety duplication of the germplasm, and the capacity sharing and capacity building, which we are doing it today. Now, we are not judging all of them. All these activities have the very well-defined procedures, standard operating procedures. But my part, I will be talking more on regeneration. That what are the principles of regeneration? The genetic basis of this? How can we really ensure the maintaining the genetic integrity of a collection? And then we'll see that how different is it what was originally from the classical genetics or the quantitative genetics or the population genetics, and how this could be really further improved using the genomics, DNA markers, and all those. When we say the general guidelines for the regeneration, in fact, when we talk of the regeneration of the germplasm, we say that maintaining the genetic integrity of the original sample is paramount. And there are basic principles when we go for the collections. There also, our first principle is to really collect the germplasm from a population, ensuring that it represents the complete diversity within that population. Because when you are going to the fields, to the farmers' fields, to the wild, a variety grown by a farmer may be segregating for several traits. It may be a combination of several types which may not be even physically visible. They may look of the same height, but they may be different for different traits. They may be different for adaptation. And basically, we want to really conserve that one. And this mechanism, the principles will vary a little bit, whether we're working with a self-pollinating crop, a cross-pollinating crop, an often cross-pollinating crop. And also whether we are talking about regeneration of a collection from the medium-term storage or the long-term conservation. So what we will define that when we have to generate those and what will be the sample size of that one. So I will be really stressing more on sample size today. So if you look into gene banks, these are the two general definitions, the two general words used, which are more or less similar, but they're a little bit different from each other. Because we have to regenerate or increase the seed of a collection under two conditions. One, if its viability has gone down from the minimum standards, or if its quantity has gone down compared to the standard limits we are setting. And the gene bank people have really tried to use these two definitions loosely, one for regeneration, when the seed viability of a collection has gone down, and multiplication when the seed quantity of a collection has gone down. For example, here I have given some examples. What is this viability? They differ with crop to crop. They differ with the storage conditions as well. For example, in general, the collections which are conserved at the medium-term storage, the minimum viability limits are 85%. And for LTS, it's 90%. For crop wild relatives, it may vary a little bit, depending upon species to species and individual genes experiences as well. But in general, if it goes below 70%, still we really need to multiply those collections. So when we are and for the seed quantity, it's a minimum number of the seeds which must be there before we go for the regeneration, because we have to ensure making these collections available to the users every time. And so when we go for the regeneration, what are the basic principles? Or what are the factors that will affect it? It may be the sample size, it may be the random genetic drift, or it may be seed viability. These are factors which will affect the genetic integrity. And these things have been very well demonstrated, worked out by our population geneticists, quantitative geneticists in the 1980s, 1990s. Good number of publications came out of that one, and especially the publications which came from J. Corosa over 2 decades. They have given us a real good understanding of what should be minimum population size for a self-pollinated crop, or a cross-pollinated crop. All those things, they're available here. The theoretical basis of those are also available in these publications. But I will not bother you for those today. I'm sure most of you are the students, And if you're still studying, you may find all these things. But what is more important here, the synthesis of these publications, which I'll go to the next slide to tell you that if we are talking about regeneration, what should be the minimum population size? So it will depend on a couple of things. One, what's the frequency of the rare allele you will perceive in a population? Is it 1%, 3%, 5%, 10%, 7%, 10%, whatever it is? And how sure you want to be that that allele, the minor allele, is retained in the population? You want to be 99% sure or you want to be 90% sure? So in any of those cases, for example, if the frequency of the rare allele is less than 1%, 1% or less, and you want to be 99% sure that this rare allele must be retained in the population, then you need to grow a minimum of 458 plants to be sure that you get it. While if the frequency of the minor alleles or the rare alleles is, let's say, 10 or less than 10, and you want to be 99% sure, then you need to grow at least 40 plants in that one. So these are the general basic scientific principles on that one. And if we look into the simple words, what a practicing breeder will require, a practicing the gene banker will require, they say a sample size of 40 individuals may be needed to preserve alleles occurring at frequencies of less than 10%. A sample of 100 individuals may be needed to preserve the allele occurring at a frequency of 5% and so. And as a thumb rule, it says that 90 to 210 seeds are needed to retain the probability of 90 to 95% alleles with a frequency of 0.003 to 0.05, and that too for a number of the loci on that one. Abhina, you have any query?
Oh, sorry, that's a mistake. Please go on.
Okay. So this is the crux. When we are regenerating the plants, we really need to know the minimum size of a population. So this was given to us based on the quantitative genetic and population genetic estimates of this one. And what ICRISAT did to see that what is the minimum size, because we needed to know the minimum size for regeneration for the crops we are working with. And we have taken 3 different crops here. The sorghum, which is primarily a self-pollinating crop, a pigeon pea, which is often cross-pollinated, and pearl millet, which is cross-pollinated. And for sorghum and pigeon pea, we took 15 individuals, measured all the traits on them, both agromorphological traits as well as we did it, genomic analysis, dot SNPs, and 25 individuals for pearl millet. Now look at this one. If you look at these figures, if we see the phenotypic basis, the diversity, the blue is the one which is a variation within accession, while as the orange is the one which shows the variation between the accessions. Now look for the sorghum. The variation within accession is also very large than the variation between the accessions. However, when we see it based on the genotypic data, it seems reverse on that one. So, because we have been looking based on the agromorphological traits or the plant traits, and we are comparing it that now what happens if the variation is known at the genic level or the allelic level. So these are the things which came to us that, okay, in often cross-pollinated crops, the variation between and within may be more or less same. But look at the pearl millet. If you see the variation within line and the variation between lines, they may be more or less the same here in this case. And this is now, we are plotting the data showing individually for 3 different crops. Look for the phenotypic data. These 15 individuals, which were analyzed individually for all the traits, most of them are really grouping in the same cluster. And the same thing is happening here when we are looking at the SNP data. While as look in the second crop pigeon P, the 15 individuals from one accession, they're not grouping together based on the agromorphological traits. While as if we look at the SNP data, most of them are clustering together. And look at the pearl millet data. Here again, the individuals within an accession are showing more variability. And as a result, we see that, let's say 15, 25 individuals, which we had taken from one population, they're grouping in different clusters. But that's not true when it goes to the genomics level. So that shows us a simple indication of this one, that analysis based on the genomics data may be more robust and more reliable than the data based on the agromorphological traits alone. And this dashboard simply shows you that how much is the variation into different things. For example, if you look at the sorghum, here, the variation between populations is higher, while as you look at the pearl millet, variation within the population is more higher than this one. And of course, pigeon pea comes in between. And looking at plotting the graphs at this one, it has given us an indication that for crops like sorghum, which is primarily a self-pollinating crop, we need at least 47 to 101 plants for regeneration. While as for pigeon pea, we may need something 77, minimum 77 plants. While a pearl millet, we will need minimum 155 plants to ensure that we retain all the plants, all the alleles of that particular of the particular crop, the particular collection of that one. So it is coming more or less the same what the thumb rule, what we have shown, what the CROSA had shown us earlier. But crop-wise, it's giving us a minimum standards, and this is what we are following here at ICRISAT. For example, for sorghum regeneration, if we have a plant population less than 47, we will not consider that population for regeneration at all. We may reject that one. And we will not go a larger population more than 100 as well, because that will be again a wastage of that one. But for pearl millet also, for example, we may need at least 155 plants. So if a population, when we are planting in the field, if it is less than 155, we will not retain that one. So having said this thing, we plant our things, and then since these crops are different, we have different mechanisms of maintaining the integrity. See, for example, sorghum. Sorghum is a self-pollinating crop. Each of those minimum 60 plants, we will bag them here, not less than what minimum size we have here. So that's a minimum one. We will bag them before the pollination, before the anthesis. And then to be sure that there are no further pathogenic or the microbial growth on these ones, we remove these bags. And then at the time of harvest, individual panicles are harvested and equal quantities of seeds are taken from all those 60 plants, is mixed, threshed together, and then bulked, of course. So that ensures us that what was our original population of sorghum, its genetic integrity will be maintained there. Look for the pearl millet, is highly cross-pollinated crops. So what we do, we plant in such a way that at any given time we have 3 to 5 ears which we can bag together, what we call a sib mating. If it's a landrace, or if it's a inbred line, then of course we sib it, we self it on that way. So depending upon the type of the population, whether we do all these things for this one. Look for an often cross-pollinated crop, which is, for example, pigeon pea. After having planted the minimum population size, which we know, and we know that the outcrossing in this is because of the insects, we do erect insect-proof net all around the field prior to flowering of this crop. And then while harvesting, we know what's the minimum number of the plants. We harvest the minimum number of the plants, take equal number of the pods from each of the plant, bulk them and thresh them and keep them in our collections. These are the self-pollinated crops like chickpea and groundnut, which do not need any intervention, but the minimum population size of these has to be ensured that that's maintained there. While as in crops like millets, small millets, which are as good as rice or wheat are, not much of the outcrossing, we do not have to take any special precaution for ensuring there, but ensuring that minimum population size, what is required for these ones, that is maintained in that one. So hence, to summarize, that when we are really looking for regeneration, we have to see what are the critical states for the regeneration and what should be minimum population size required for that one, and what are the interventions to ensure that there is no outcrossing with other accessions at all. So with this one, I will stop here and thank you very much. I will be available for all any questions whatsoever. And I will request my colleague Damaris, our floor is to Clinton now.
Okay, thank you so much, Kuldeep, for that awesome presentation. I believe it's Marie-Noel who will introduce the next speaker.
Okay, thank you very much, Kuldeep, for the presentation. Let us now give the floor to Vetri and Damaris. I don't know who, is Damaris? Okay, Damaris, you have the floor. Thank you.
Hi, hello everybody. Sorry, I am trying to drag this thing out of my screen. I hope you can all see my screen, the right screen. Is it the presenter's mode or I need to swap my display?
Yeah, it's good. It's good. It's on presentation mode. Yeah,
okay.
It's good.
So I'm relatively new to the GeneBank community. I'm Damaris Odeny. I work at ICRISAT as genomic scientist, molecular breeder, but I do provide a lot of support to our GeneBank in terms of molecular characterization. My task today is to just elaborate in a few slides the use of DNA markers for diversity and structure analysis. I'm going to borrow from experiences working together with the GeneBank, but also a few others, broadly on some of the things that we've done before. So my presentation will build on some of the presentations we had yesterday, both from Ranjana, Carolina, and Zach. And you already, got a clear understanding on, sequencing and generation of SNPs. Now, diversity analysis can be done using different kinds of markers, all sorts of markers that you are aware of, all sorts of molecular markers you're aware of can be used for diversity analysis. Simple sequence repeat markers have been used extensively. However, for the sake of this presentation, I will restrict it to SNPs because I do know that there are very few people who are still using simple sequence repeat markers, but also the future is SNPs, so it's just easier to give examples from SNPs, and it needs no introduction after the talk we had yesterday. Now, when it comes to SNPs itself, there are different kinds of SNPs, or there are different ways to generate SNPs. So the consideration one has to give in terms of how you're going to generate the SNPs you're working with depends on a lot of things. So it depends on the crop in question. So for example, if you're working with a hexaploid or a polyploid, sometimes it's easier to just use SNPs that have already been pre-designed, because calling the SNPs afresh it's a lot of work to be able to get it right when you're working with a polyploid crop. You also need to just figure out, how well are the markers you're going to use distributed across the genome? Because if you're doing a genome-wide diversity analysis, you really want something that is representative of the genome. And as I was saying, you also need to think about the ploidy, how the levels of heterozygosity. If you're working with a crop that is highly heterozygous, you have a problem because if you're using SNPs that are developed from highly heterozygous region, then you may not be able to call the SNPs correctly. There are so many considerations that you have to make to help you decide the type of SNP markers you're going to use. And in the next slide, I'm just showing why one would want to do genetic diversity analysis. Sometimes, especially with respect to the gene bank situation, most of the time you want to understand the true extent of differences, especially let's say you've seen some differences in the field, you want to see if those differences are actually genetic. Sometimes you want, maybe as a breeder, you want to select the most diverse parents to optimize heterosis depending on the crop you're working with. You also may want to remove redundancies, and I think Vetri is going to talk about this. Also, sometimes you just want to develop a reference set, which my colleague Vetri will talk about, but also maybe to confirm relatedness. But in many cases, actually, as students who are learning, sometimes you're doing it to publish an article because you are required to publish to graduate. So sometimes you're doing it to learn and publish an article, right? So there are so many reasons why you'd want to do a genetic diversity analysis. Now, this slide is just showing the three main ways that SNPs are generated. The whole genome sequencing approach, which Ranjan attached yesterday, where in this case, you're actually doing complete genome sequences of individuals you've selected. This is mostly practical if you're working with specifically breeding lines and you have maybe 5-10 breeding lines for which are representative of, let's say, different breeding programs, and you're trying to develop markers across those 4 main or 10 main. It's not a huge number, but you want to capture every little variation that there could be and develop maybe a panel that you can use to eventually characterize the progenies or subsequent breeding lines you generate from there. And so it's rather expensive. It's also capital intensive because you're trying to capture the whole genome. Now, the most common one, and the one that I think is relevant to this panel is especially complexity reduction sequencing. And this is where you take your genotypes that you want to genotype and digest, or just cut the DNA into random pieces and then try to put them back all together in a way that you can predict the regions that are going to align together and help you to call SNPs. Now, one of the ones that you might be familiar with, which has become really common now, is DART sequencing. So basically what DART sequencing does is it generates very low coverage sequencing of your target genotypes, and then put them together and call SNPs, and then use that data to characterize your germplasm. It is relatively affordable because you don't have to capture every part of the genome, like the whole genome sequencing, but it still gives you pretty much good information on SNPs, especially if you're working with a diploid crop. It gets a little bit complicated if the crop you're working with is a polyploid, but we can come back to that later. Now, another one that is many times I think is overlooked, but is especially really useful, is the use of existing panels. For diversity analysis, existing panels work absolutely fine. And especially if you have existing panels that have been characterized and defined, maybe there's top 10 most informative markers for cassava. Those would work absolutely well. It's affordable, it's easy, the data sets are easy to handle, and it does the diversity analysis just fine. And you can have existing panels at very low density. You can have just maybe 10 most informative, you can have 50, you can have 100. It also depends on the genome size that you're working with. But you can also have some that are medium panel, up to 10,000 markers, but you also have some that are high density, maybe up to 100K. I think in wheat, we even have 1 million SNP chips. So it really depends. But for diversity analysis, you really don't need that massive number of markers. As long as you have markers that are well distributed across the genome and they're informative, you're good to go. Okay, so now you've got you've generated your data set, especially when you're working with reduced representation of the genome or whole genome sequencing. Once you've generated your markers, one of the most important things that you have to do with that is to filter the markers. If you're working with markers that are not predefined in panels, then you have to go through the pain of trying to understand how well or how informative those markers are. And some of the obvious things that I used to filter that marker to make sure that you improve its informativeness is to filter for minor allele frequency. And this is basically removing markers that are likely to be there by chance. They're most likely sequencing error. However, when you're working with GenBank material, one needs to handle this a little bit more carefully, because GenBank material also has diverse material that in some cases, they actually do have minor alleles. So you can have collections you've made from all over the world, and there's an allele that exists just in 5 individuals, maybe one from Africa, another one from Asia. So this is one of the things that you really need to be careful. However, if you've done whole genome sequencing where you're very confident with the coverage of your samples, you can retain, you don't have to do minor allele frequency, especially if you're working with GenBank material, which you suspect that there are very rare alleles there, there could be very private alleles there. So what you do in that case, then you do your sequencing at a higher coverage, so that when you call the SNP, even though it's just 5 alleles that are present versus 100 of others, you can still retain it, so you don't have to filter it out. However, if you're working with improved material, then when you only find that this allele is only existing in 1 or 2 individuals, then it's probably a sequencing error. So you can delete it. You can remove it. But the other thing that you tend to do with SNPs that you've just generated and you want to use for diversity analysis is just to do a quick polymorphism information content analysis to be able to see how much those SNPs are able to differentiate genotype 1 from genotype 2 or different genotypes. And usually, this is one that we do like as a standard. When we generate the new markers, we filter them, we've looked at polymorphism information content. You can see the difference. Before filtering, you can find that, and one of the most important is the PIC, the polymorphism information content. You see here, after filtering, you increase it to like 0.31. Before filtering, it was only 0.29. But the example I'm giving you is actually from a really good data set. In most traditional data sets that you've just generated, you will find maybe your markers mean polymorphism information content can be as low as even 0.1. So you want to increase it, because polymorphism information content also shows you the informativeness of those markers. So there's confidence in knowing that your markers are informative, because that's what's going to differentiate your genotypes from the other in a way that is going to be useful in your breeding program. So anyway, so that's the first step that you will really need to do, especially when you've just generated new markers yourself. And this applies also if you've just generated sample sequence repeat markers or whichever other markers. It's just that for this, SNPs have just become so relevant and the focus is on them. Now, one of the ways that you can just do a quick check to see that your markers are actually informative is test them across your population and see to what extent they are actually explaining the genetic variation within your set. Now, I'm just giving an example here of SNPs that we used in a panel for groundnut. So in this case, we looked at 150 SNPs that were existing in a panel. And for this population, which was around 384, the 150 were explaining 36.5% in the first two principal components when we did the PCA analysis, which is pretty good. This is not bad at all. But just to show that when you filter and take the most informative, the more informative SNPs you retain for your set, the more they are able to explain genetic variation better. So for example, you see now when we narrow that down and we take just the top 10 most informative, by the way, now what this means is that those top 10 are able to differentiate 64% of the time when you choose when you want to differentiate a random genotype here from the other, 64% of the time, those 10 SNPs will be able to do it. And you can see the more you do that, the more you are able to even group them even better. And so informativeness of markers is absolutely critical when you're trying to use them for diversity analysis. Now, this is a good an example of a marker set, and I have to also put a disclaimer that this marker set was already pre-designed. So it was already existing in a panel. And that's the good thing, that, if you're using markers that are already existing in a panel, someone most likely already selected markers that were informative and retained them. So this one, as you can see, we used these 7,523 high quality markers that were already existing and used them across about 200 groundnut germplasm. And you can see those markers were explaining 67.3% genetic variation within that germplasm, which is pretty good. And you can actually see when you look, it's already clustering the different classes together. It's not very difficult for you to try and see where are the clusters. Now, that's the other thing, when you're working with markers that are not informative and you use them for clustering, it gets very difficult to figure out where the clusters are, just because the markers are not that informative. In fact, I can give an example here. This is probably not a very good example, because 36.5% of the time, if you're able to distinguish your genotypes, is not bad. But you can see, in this case, the groups are not as well defined as when you have, in this case, 64% genetic variation explained, or 49.9% genetic variation explained. And as you can see, the same markers, if you do use them to draw a dendrogram, you can actually clearly see, the clusters. Here you get the cluster for Virginia, class of groundnut. Here you can see clearly. And granted, in this case, you can actually use this because you know that the clustering is so good. You can see, there was probably 2 or 3 here that are put in Spanish cluster, but they belong to Virginia. So you can remove them and so forth. So yes, you really do need to always use a good informative set of markers. So what's the practical application of this kind of analysis? And yeah, I'm going to try and finish what I'm left with in 5 minutes because I need to give time to my colleague Vetri to finish. Some of the practical applications is, if you see in this screen, we have, and I'm giving this example for sorghum. Now, we have sorghum classified in different races. The main races are bicolor, Dura, Guinea, Kafir. But we also have some other smaller races. And now because sorghum is outcrossing, now we also end up with, many interspecific races. Now we have also bicolor Codetum bicolor, Codetum Dura. So you just have, you end up with so many categories. Now, what markers, informative markers will do for you is to help you group them in a way that you can actually enhance the grouping. So instead of having all these many, you can then condense it so that it's easier for someone to separate. And you can see this is an analysis we just did this week with about 1,200 sorghum accessions using publicly available databases. Data sets, and you can see how beautifully these have been classified, and so we can actually condense this long list of races and inter-races together and say, okay, this bicolor Codetum bicolor cluster has all these, and this is to what extent they are explained. Now, another example I'm going to give, to be able to do correct classification. So if you see this table that I just talked about for sorghum, we have 148 in that list where we don't know where to classify them. Now, I'm going to give you an example. In this particular case, after we did that diversity analysis, we can go back and see, okay, so for example, I hope you're able to see my screen where my arrow is. There's unidentified. So it has UNID. We have an unidentified genotype there that is clustering together with Duras and anything that has Dura hybrid. So then we can say most likely it is a Dura. But the good thing with the gene bank is, you give that classification specifically to the set you're working with, because in the field, it might be a very, very different thing. So you would have to then go back and look if it actually behaves the same way in the field. However, in this case, we would just treat it as a contamination because these were things that we got in public databases. It's possible that if we go back to the gene bank and we get the right ID or the right genotype or the right seed for this IS, we may actually find that clustering in the right place. So sometimes it's just correct classification or removing contamination. Again, you can resolve misclassification. You can use it to create core sets, and this is one of the ones that I was going to give, where about 1,049 genotypes, we did diversity analysis and were able to come up with a core set of 205 groundnut data set, which is in the groundnut improvement for the Groundnut Improvement Network of Africa. And that's the same way also we come up with core sets, and I think Vetri will talk about that. I just want to touch on structure analysis really, really quick before I wrap up. Now, what is the difference between structure analysis and diversity? Why do we need to do them? Most of the times they're probably even explaining the same thing. However, structure analysis then now help you to create strategic groups. So now if we go back to what I was talking about here, and now you end up with all that huge list. Now, when you come to doing structure analysis, it helps you now to classify them, even though they are in small patches, you're able to put them together in defined groups. Now, there are two or three major software that I use for structure analysis. The traditional one was actually software called Structure, but it was actually quite computationally demanding. Now we have discriminant analysis of principal components. And basically what DAPC has done is to be able to add between groups distinction from principal component analysis. When you just do principal component analysis, you just have all these clustered individuals. Basically, what principal component analysis does for you is detect variation between genotypes. But when you add the discriminant analysis component, it will then look at specific groups that share alleles and put them together. And then there's a way that you can actually go back and look at the optimum numbers of populations and then use it to create different subgroups that suit your case. Now, there's so many ways that you're able to apply that. As I've indicated, it helps you to structure, put some kind of grouping when you have it all over the place. You can also optimize your core sets. You may have generated core sets, but you want to generate another set, so you can start with the larger one, but you want to create another mini core and then another mini core. So it helps you through groups to be able to identify that you actually have captured the different sets. Sometimes also the groupings can show specific enrichment for certain traits. So if you're trying to distribute material, let's say to turkey that are disease resistant, you know that materials that are within a certain grouping have disease resistance, for example. So it helps you to utilize it in that way. So I'll end there and give over to Vetri, and my sincere apologies, Vetri. I think I've run over time a little bit. Over to you. Sorry.
Yeah, thank you, Damaris. Shall I start? Allow me to share my screen. Okay. I hope the slide is in the presentation mode. So I myself, Vetriventhan, genetic resources scientist working with the ICRISAT Gene Bank, India. So I will be talking on Germplasm diversity subset. So let us go directly to the topic subject. Okay. So why we need a Germplasm subset? So we need a Germplasm subset to enhance the use of Germplasm diversity in the crop improvement. That is the purpose why we need a Germplasm subset. But developing Germplasm subset depends on the purpose. What is the purpose of the developing subset? Do you need to capture maximum diversity of a collection that are conserved in the gene bank? Then we may need to go for a developing core collection or mini core collection. Or do you need to identify a trait-specific subset using the climate variables and then rainfall, temperature, and other soil variables? Then the approach is called focused germplasm identification strategy. I think this was presented very well yesterday, so I will not go deep into this fixed strategy subset. So I will talk more largely on core collection and mini core collection only. Okay, if you come to the Germplasm global status, you have almost 7.4 million Germplasm accessions are there, which are conserved in more than 1715 gene bank. And there are about 25 to 30% unique Germplasms are there. And these are all the basic information the students and other early researchers may be interested to know. And this database is something like where you can see the PGR database in the few views. World Information and the Early Warning System on Plant Genetic Resources for Food and Agriculture, where you can see the region-wise accession, number of accession from different genera and species, and number of gene bank. So this also you can get information from this database. So ultimately, we have large number of Germplasm in many gene banks like the National Gene Bank and different gene bank in the university or different places. For example, at ICRISAT, we have almost 132,000 Germplasm accessions of 11 crops. By crop, If you see example, sorghum, we have more than 44,000 germplasm accessions are there, and palm nut, more than 25,000 germplasms are there. Similarly, chickpea, pigeon pea, groundnut, we have global collection of these crops accessions. But how to enhance the use of this diversity in the crop improvement? That is the important thing. That's why we are conserving, and we wanted to utilize this germplasm diversity in the crop improvement. Basically, what we will do, we will be doing germplasm characterization for descriptor traits. That is the basic things that we do whenever we receive germplasm. This will be the information that will be added to the database. In addition, we can do germplasm evaluation. The issue is when size of the collection is large, it is difficult for phenotypic and genomic characterization, and also it demands more manpower and resources also, because it is difficult for the systematic and multi-location and replicated trial of a large number of germplasm is always difficult. Nowadays, we have high throughput phenotyping and genotyping facilities are there. Now we are trying to explore those options also. So coming to develop this core collection concept, the core collection concept was proposed in 1984 by Frankel. It represents with a minimum of repetitiveness the genetic diversity of a crop species and its relatives. So which data you can use for developing this subset, core subset? So you can use passport data and taxonomical data, and also if you have phenotypic data and genotypic data also. All this data, you can use it for developing subset. So the subset core collection is like, it is 10% of the total collection of a given crop species. And sometime the core collection itself a little large in size. You take example of sorghum at ICRISAT, almost 40,000 germplasm is there. And if it is 20%, 10% of the total collection, it means 4,000 will come. Currently, the core collection of sorghum is 2,247 accessions are there. So this core collection itself sometimes large in size for multi-location and systematic replicated evaluation of this core set. Therefore, the concept called mini core collection came, which is core of core. The mini core collection is 1% of the total collection and 10% of the core collection. So the core collection and the mini core collections came into the approach came into practice. Here, this is the methodology how we can develop the core and mini core collection. So when you have the germplasm, which is 5,000, 10,000 germplasm is there, and then we will be using the passport characterization and the statistical method. We will be developing the core collection, which represent almost 10% of the total collection. And then if that core collection itself little large in size, then we may need to go to the next level, which is called mini core collection. So the mini core collection is 1% of the total collection and 10% of the core collection. So here shows the methodology how to develop the core collection. So first, if you have the 5,000 germplasm or 10,000 germplasm, and you can group the germplasm into different way, like taxonomical grouping you can do, or regional grouping you can do, or based on any other data, similarity data, you can group the germplasm. This is like you see here, group 1, group 2, group 3, like that. Then do all the statistical analysis and do clustering. After clustering, from each cluster, select 10% of the accession to make the other core collection. So this is the approach. And then the same approach is followed to develop mini core collection also. See, there are many software tools are available nowadays, and you can use for developing core or mini core collection. If you want to develop a core collection using phenotypic data, there are R packages are there, CC Chooser, which is for the phenotypic data. Or if you want to do with the genomic data, then recently one publication came, Shiny Core R-based program is there. And also another SNP-based program, R-based program is called GenoCore. You can use this the packages for R-based packages for developing core collection. In these packages itself, if you go explore these packages, R packages, and you can see the input file format and then the other different methodologies, and then it will ultimately give you the core and mini core collections. And if you have the both phenotypic and genomic data, then you can also use this core hunter. Now the version 3 is there. So this is something I just wanted to tell. There are different software tools which is available for developing core collection. This is the page where you can see the core chooser is there. And here it uses agronomic data, morphological data, and phenological data. And from this place, you can explore more on about this application. And another one is Shiny Core, which is R-based program for establishing core collection using SNP data. So this publication, if you want to understand more information on this, you can also go through this publication. And another one is like GenoCore that I already explained. Here, Python-based coding also there, and then R-based packages also there. Based on your expertise, you can always choose the right best one. And this is the core hunter, where this is used for developing core collection using both phenotypic data as well as the molecular data or SNP data. For the genotypic data, they use the modified Rogers distance method, and then for phenotypic data, they use the Covers distance method for developing the distance matrix, followed by developing the core collection. And there are many core collections developed in different crops, starting from rice, sorghum, groundnut, and many other crops. You can see there are a number of core collections are there. And also using either phenotypic data or genomic data, or both phenotypic and genomic data. For example, here the 38 SSR markers are used to develop core collection. So, and then here both for rice, mini-core collection was established using 26 phenotypic data and then 70 molecular data, 70 molecular marker data also like that. So one can choose it's always better if you have the data of phenotypic as well as marker data, then it's good to go with both, using both the information. But if it is large collection is there and then genotyping of the total germplasm is difficult, maybe the passport and the characterization data also sufficient to develop a core collection. So here, so after following this approach of developing core and mini-core collection, so ICRISAT, we have developed a core collection and then mini-core collections for all our crops we are working. So you can see all the crops we have core collection also. The core collection is starting from 75 or 56 accessions in little millet to 2,246 accession in sorghum. So when the size of the collection is large, then we try to develop mini-core collection also. The mini-core collection is starting from 35 accession in foxtail millet to 242 accession in sorghum. So this is a small subset that can be very well used in multiple location replicated trial for different traits also. So I will take one example of sorghum. Once you develop the core collection or mini core collection or a diversity subset, then what is next? So for example, in sorghum, we have core collection of 2,246 accessions are there. The mini core collection is about 242 accession. In addition, we have the composite collection is there, reference set also there. So these are all the subset that we have developed in sorghum. Similarly, in other crops also, I am here trying to present related to sorghum. So what is next? Once you develop the core collection, it's not that our work is not ending there. So we need to work further to evaluate the germplasm for different traits. The core collection, the key difference between the diversity subset and then focused germplasm identification strategy is the core or the diversity subset, you can look for different traits, many multi-traits. This is for the multi-traits, but in case of focused germplasm identification strategy, that is developed for a certain trait. So you cannot explore multiple traits in the focused germplasm identification strategy, but in the core collection, you can evaluate the germplasm for multiple traits and identify germplasm sources. See example here. In sorghum, 44,000 germplasm accessions are there, which are originating from 93 countries. And then core collection, mini core collections also there. And by evaluating these core and mini core collection for grain nutrients, we were able to identify the accession which are rich in iron, zinc, and also accession which are rich in iron and zinc also. And some accessions are superior in terms of grain yield also, in addition to the grain nutrients. So this is how we are able to identify the trait-specific sources in using the core or mini-core collection. Not only that, we try to assess the photoperiod sensitivity, photoperiod and temperature sensitivity of sorghum, and then we planted them in the long day and short day conditions, and then we tried to classify the germplasm into photoperiod and temperature sensitivity, photoperiod temperature insensitive, so that based on the requirements, the breeders can use a different type of material for developing the cultivars. So there are almost 18 accessions which showed photoperiod and temperature insensitive. So these 18 accessions are much useful for the breeding the diverse, very well adapting germplasm, the cultivars. So coming to not only that, but we also assessed this diversity subset for abiotic stresses like post-flowering drought tolerance and low temperature stress tolerance, and then identified several germplasm sources for this triad. And also, and then we assessed this germplasm for the biotic stresses also, like grain mold and then downy mildew. So for grain mold, we have identified 50 accessions which are sources for this resistance. And then for downy mildew, 6 accessions are resistant to downy mildew. And then similarly for the foliar diseases also, like anthracnose, leaf blight, and rust also, we were able to identify resistant sources. This is the importance of the diversity subset. Once you have the small diversity subset, then you can assess that subset for n number of traits and then identify trait-specific sources. And here, finally, we try to identify multi-trait specific sources. Like, for example, this accession like 4698, which is having high bricks also and then resistant to spotted stem borer and shoot fly and aphids. Similarly, other accessions like it is having large seeds, high bricks content, having post-flowering drought tolerance and charcoal drought resistance also. Like that, these are all the accessions which are having multiple traits and also having considerably good high yielding background also. And interestingly, if you see the right side, the resistance to multiple diseases. So there are accessions which are resistant to multiple diseases. You see here the 473, which is for the grain mold, anthracnose, leaf blight, and rust. For both 4 diseases, this accession is resistant. Similarly, the other accession, 2 accessions which are resistant to anthracnose, leaf blight, and rust. So this is something which is very much interesting. One can transfer this trait from this accession itself. Further, we try to identify most diverse accession, which are also sources for multiple traits. See here, and then the accession in the left side, and then accession in the middle. These both are highly diverse accession with the genetic distance of 0.478, and also sources for multiple traits. When we cross these two things, we are not only bringing the diversity, but also bringing the traits also with this. So it will also have the more heterosis also. And then here, not only the core collection used for the identification of trait-specific sources, but this core collection or mini core collections are very good association mapping panel for genomoid association studies also. So we took a sorghum mini core collection, and then this sorghum mini core collection was genotyped for different markers. Initially, we did with the SSR markers, later with the SNP markers. And then we tried to identify marker trait association for key traits, like starting from plant height to maturity, and then diseases also like grain mold and rust resistant, anthracnose resistant, like that. So this is like not only for identification, but also you can use it in the genomic studies also. So ultimately, the core collection or mini core collections are important for identification of trait-specific sources and also their utilization in the crop improvement. So in the last 3 presentations, we were discussing largely on conservation, particularly regeneration importance, and then diversity analysis, and then diversity subset development. So I tried to finish it within time. With this, I'm concluding my presentation. So,
Okay.
thank you. Thank you.
Thank you so much, Vetri, for that wonderful presentation. Before we move to the Q&A, we've been required to take a group photo. As much as possible, we want you to turn on your camera and we'll quickly take a snapshot of everyone present. It's going to take a minute to enable the camera of most participants, but yeah. So please. If your camera is enabled, please turn on your camera as we're about to take a group photo.
So was it a hectic week for you, Marie-Noël? Are you enjoyed it?
You are muted.
Okay. I said everything's fine, yes. Clinton, please just do what you can and then so that we can move on, please. Thank you.
Okay, thank you very much. Because of the number of participants, I am not able to enable all. So we'll quickly take the snapshot now. I think we have about 10 to 15 people enabled. Okay. Okay, thank you all. I've been able to screenshot and we will move on to the Q&A. So you can now turn off your camera if you're not speaking. Everyone is invited to ask question by raising up their hand and then putting the questions up in the Q&A. The first person is Elizabeth Johnson. Hello, Elizabeth. Can you ask your question now? Kindly unmute.
Good morning, colleagues. Well, I'm in Barbados, so good morning, good afternoon to you all. Thank you so much to the ICRISAT team for those presentations. And yes, I am not camera ready this morning, so I did not turn on my camera. But I wanted to ask two questions, one to Kuldeep, and this is actually more out of curiosity than anything else. With the pigeon peas that's actually cross-pollinated and insect pollinated at that, when you put the cages over, are you actually hand pollinating each one in the cage to generate the seed? It's really just out of curiosity on that. And the other one I think is for Damaris and maybe Vitri, who was last. And I think Vitri may have clarified this a little bit. When you are constructing your core collections from the bigger subset, how much are you interacting and taking the information and recommendations from the breeders or the people who have done the morphological descriptors to ensure yes, I know the statistics looks good with the SNPs, and we're making sure that we are getting genome coverage, et cetera, et cetera. But the core, one of the core reasons for having a collection is to ensure that you conserve genes that will be useful for things that we have not encountered yet or that we don't know about. It's this futuristic vision. So how are you ensuring that within the diversity analyses that and the markers and the development of these core collections, that we're actually maintaining this vision of, keeping things that we may encounter in the future? Thank you very much.
Well, thank you, Elizabeth. For pigeon pea is naturally a self-pollinating crop.
Okay.
However, if the insect populations are present, then insect visit different flowers, collecting pollen, moving to the other ones. And when we are having a net around it, there's no insect population. So the population is then in generally self-pollinating. We don't need to do any selfing artificially in pigeon pea. I hope that's clear.
Yes, thank you for that, Kuldeep. I think I missed that part, that it was a self-pollinating crop. Thank you.
Yes. So regarding this core collection, how will we capture the traits information which is needed for the breeders? So actually, when we develop core collection, when anyone develop core collection, it depends on what type of data you have. Whether you have only the characterization or the descriptor traits only, or you have additional information like genotyping data and then trait data. So all those things also. So by using that information, when you develop a core collection, then maybe you can also capture the traits that we are looking also. But basically, core collection is to capture the diversity. And if you found one or two traits out of that total subset, if you want additional source of that trait, then you can always go back to the core collection, or you can always go back to the total collection based on the cluster from which the particular accession was selected. So that way we can always explore. Is that fine? I addressed your question.
Okay. Yeah. Okay. I guess. So all right. Yeah. I guess that can do. Thank you. Thank you.
Okay, thank you so much for that. We have another hand up. I think Marie-Noel has a question. Masood, sorry. Masood. Hello, Masood. Can you unmute and ask your question? Oh,
Hello.
Masood, okay.
Do you have my
Yeah, we
voice?
can hear you. Yeah, we can
Yeah.
hear you.
It seems that my voice is not reaching there.
No, we can hear you. Go ahead. We
Okay.
can hear you.
Let's consider a cross-pollinated crop such as maize or sunflower. How we can use the information from the core collection to produce the best hybrids regarding, let's say, the yield or something else. I mean, how we can compare or how we can use the information from the core collections for producing or developing new hybrids. Can you give me an idea in this regard?
Yeah. So the core collection itself is the miniature of the diverse germplasm. And in one of my presentation, I tried to show most diverse succession with the traits, multiple traits. So if you combine those two most diverse succession in your crossing program, then ideally you will be bringing trait as well as the diversity also. That's how you can breed the good cultivars.
Yes, that's true. But when we cross two diverse, let's say, accessions from two different core collections, the other parameters also can become out. Let's say the resistance to the biotic stresses and abiotic stresses. I mean, it's a combination of different traits. How we can consider these several traits in producing a hybrid, taking advantage of the core collections. My question is clear.
Yeah, let me respond, Vetri.
Yes, yes,
Yeah,
yes.
thank you, Masood. I think your question, if you address it to Damaris, that how the clustering, the diversity analysis can help you more. In fact, it's the diversity of the whole collections, what Damaris has shown, how can you generate it. That will help you in really identifying the exceptions that you can really put in different heterotic groups if you have that sort of information available in that case. Cores per se may not help you. Cores are diversity analysis. Now, to which of the, if you have a heterotic pools, now to which of the heterotic pool it will go, it may not be clear from the from the core. But definitely, if you are using the core and really making again the clusters or the diversity analysis of that one, that can help you in identifying the accessions or germplasm that can go each of the heterotic pool, which in turn you can really use for generating inbreds for developing the hybrids. Maybe if Damaris wants to add something more to this one, Damaris?
No, I think you captured. Yeah, I think you captured it well. Thanks.
Okay, Masood, that ends the question. In the meantime, we've experienced some glitches with the Q&A and the chat. So please, if you're unable to raise your hand, you could send me your questions by chat. So we have a question from Kandelem Ilri. He's asking, he said, we experienced a peel millet during multiplication time and that have not set seed. How you confirmed and produced it. During characterization, how we keep and characterize their seeds color as peel millet is outcrossing crop. Does that need bagging or not? So this is a question from Kandem Erie. I don't know. Did you get the question?
Yeah, please.
Do I repeat the question again?
Yeah. So whose question is this one? Because it has not gone to the serial wise. I was looking into Q&A sessions. So who has asked this question? Let me read that one.
Okay. So the question is from Erie, and for some reason the Q&A is not working today. So they sent me the question to my chat, and I'm trying to read it out for you.
Yeah, please.
Do I take it again? Okay. So he's asking, I think he said he experienced a peel millet during multiplication time that have not set seed. He's asking, how did you confirm and produce it? And then he says, during characterization, how do we keep and characterize our seeds color as peel millet?
So,
It's
Vetri, you will
crossing
respond to that question?
Yes,
Yeah.
yes.
Yeah, respond to that question.
So the palm millet, actually, you need to plant it in appropriate season so that the whatever photoperiod temperature sensitive germplasms are there, then it will flower and produce seed. So we need to choose the right season for planting. So that is one thing. And another thing, during characterization, I think he is asking whether we can do bagging or we can do the regeneration, something like that, I assume. When you do characterization, we characterize it under the natural condition and we do not bag it. But for the regeneration, we bag it, cluster bagging, other things. So characterization is the separate activity and then regeneration is separate activity. So for regeneration, you need a minimum sample size also, and then you need to do bagging also. So that way it is different. So for characterization, you can keep the panicles open and then allow it to set seed, and then so that you can characterize the flowering time and then seed color, panicle length, width, all those things you can do. When you do bagging, then it's difficult. I hope I
Okay,
answered that question.
Kendall, if you can hear me, does that answer your question?
Yes. Avina, the Tina is asking, Tina has raised his hands.
Yes, yes. Before Tina, we have another question on the chat. It says, I would like more information about the Core Hunter software. If you have used it before, how does it work? Does it try to correlate traits with genotypes? For example, imagine I have 10 phenotypic data points for sessions, along with DATSEC data. Can I group my sessions based on combined phenotypic and genomic data? Thank you. So that's another question before we take from Tina. Over to you.
Yeah, that core hunter is used for developing core collection using both phenotypic and genotypic data. So you can develop a core collection by combining these two. It will initially it will develop a distance matrix of both the phenotypic and then genotypic data, followed by the clustering and then identification of the subset.
And that's what is referred to as composite cores, which uses both the genotypic data as well as the descriptor data.
Yes.
That's great. So once again, before we hand over to Tina, I would like to encourage us to raise your hands if you have a question. If you're unable to speak, write to me on my chat. So over to you, Tina. Can you unmute and ask your question?
Yes. Hi, can you hear me?
Yes, yes.
I can hear you.
Great. So I have a question for Damaris. So Damaris, during your presentation, you mentioned polyploids, which usually make genotyping difficult. So I would like to ask you if you can a little bit elaborate on that, because I work also with polyploids, mainly with fruit species, with fruit trees, and and up until now, we were working mainly with SSRs, but I would like to broaden this, maybe try some new tools and things like that. So maybe if you have advice.
And what's the ploidy?
Oh, yeah, I work with plums, which are hexaploids, but a lot of times I work with different ploidy levels. So they can be either diploids, tetraploids, or hexaploids, and so on. Yeah.
Okay. So the only difficulty you have when you have higher ploidy numbers is that you need to generate a lot of data to be confident with the SNP markers that you're working on. You see, for example, if you were just working with a diploid, if you generated SNP data through sequencing, you probably just need 4 times coverage to be able to call the SNP correctly, because the way that you call the SNP correctly is a game of numbers. So if you have 2 sequences aligning in that region with allele X and another 3 with allele Y, you're confident that that's a single nucleotide polymorphism area, right? So if it's a diploid, you only need like 5 times coverage in that area to be able to call the SNP correctly. However, if you have a hexaploid, remember the different combinations for each of the subgenomes. So it's almost 2 times 6, 12 times. So you need for at least 12 times coverage for each of the subgenomes to be able to call the SNPs correctly. And that's why when you're working with polyploids, it's always recommended that the markers are pre-developed so that you can use it routinely. Because now when you pre-develop, you pull effort so that if you're sequencing, let's say, the representative 100 genotypes for that particular species, you're doing coverage of at least 100 times to enable you to call the SNPs correctly, and then you array them on a panel, and then in future you just use the ones that you have determined and established without a doubt that these are nucleotide polymorphisms that will define or help differentiate your genotypes. But if you just did a random low coverage for polyploids, most likely you will be working with homologous SNPs, which are SNPs that are just shown between the 2 subgenomes of the same individuals. Remember, you want to differentiate individual 1 from individual B, not just looking at how different they are within their different subgenomes, which is a completely different study. And because the sequencing costs now are generally low, you should be able to shift from your simple sequence repeats to SNPs, but the initial investment might be a little bit heavier. I know crops where simple sequence repeats are still the markers of choice, just because it will otherwise be so expensive to shift to SNPs because of the ploidy nature. So we can have another side chat. You can send me an email specifically, and we can design something if you need to. But yes, I think those are some of the options that you have.
Thank you so much for your kind answer. Thank you.
Okay, thank you so much also for that. There's another question on my, the question is from Mahamadi. Mahamadi is asking, does the principle of co-collection deals with heterosis? Does the principle of co-collection deal with heterosis? That's a question from Mahamadi. What to you?
Yeah, I didn't get the question. Can you repeat?
Does the principle of co-collection deal with heterosis? I think the group chat is working, so I will just paste the question on the group chat so you could see.
Yeah.
Okay.
I'm trying to see.
The answer is no. I mean, the core collection does not work with the principle. Heterosis works with the principle of the diversity. So the core collection is a represents overall diversity, but within that one, which will be the accessions, which will be heterotic with each other, that will not be determined until you do the diversity analysis.
That is great. Mamadi, does that answer your question? Okay. Before we go back to the charts, we have a hand from Martha. Martha, can you unmute and ask your question?
Okay, thank you for the great presentation for all the presenters today. My question is related, I have two questions. First of all, one is related to heterosis. For diploid species such as cowpea, in our gene bank, I've been working with cowpea. So I was wondering how could we try to assess the level of heterosis on the core collection that we've been working on? The second question is related to the quality of markers, SNP markers that we could use, because we have tried to come up with a core collection in which we did the sequencing by genotyping using a DDRAD method, but we didn't get the coverage. So I'm not sure about the quality of these SNPs. Is the number of SNPs related to the quality? How could I assess the correct markers to use for the further analysis that I intend to do? Thank you.
So I can take this. Your first question. So if you're asking about heterosis in cowpea, I'm not too sure that you. Okay, so heterosis and core collection is not related at all. So these are two different things. But again, in cowpea, I believe is highly inbred. Now, if your question about the number of markers, now remember the number of markers that you generate from sequencing has nothing to do with the quality of the markers. You can actually generate millions of SNPs that are all terrible quality. So the way that you improve the quality of the SNPs is in enhancing the coverage of how much sequencing you generate for each of your individual samples. So DDRAD, unfortunately, is very low coverage, and also depending on how the libraries were prepared, you can end up with only maybe less than 0.5 times coverage. That is not a bad coverage for a crop such as cowpea because it's inbred. Chances are most of the loci are fixed. And so in an experiment where you've done sequencing for, let's say, 200, you might find 5 genotypes that have the same allele. When you have more than 5 alleles for that particular locus, you can still call the SNP correctly. However, if you've ended up with just one coverage for one allele, you can't call the SNPs correctly. So you don't have enough layers at one locus to be able to say, yes, without a doubt, what I'm calling is not sequencing error, but it's a SNP. It's actually a difference between different individuals. And so that's why I was talking about filtering. When you're doing filtering, you're removing SNPs that may have occurred due to error. And most of the software that we use will let you know if the coverage is just 1 or 2, and you only have 2 nucleotides there that can be called, it will throw it out as poor quality, and so you cannot go with it. So if you generated 1 million SNPs and they're all bad quality, unfortunately, then you cannot use them. But the best thing that you can do when you generate something like that is to do a stringent filtering. And if the objective is just to do diversity analysis, it's better to retain 10 good SNPs that are of high quality than retain 100,000 that are all terrible quality, because then they cannot really help inform your analysis in any way. So I hope that answers your question. And I can let somebody else maybe address the heterosis question.
Well, thank you so much, Damaris. I don't know if you can also share some software that could be used for filtering. And I'll also be happy if you could share your email so that I can make follow-up later to discuss further about this. Thank you.
Thank you.
So, Vetri, you respond to the question of heterosis, or?
Actually, Damaris already mentioned that.
I think the answer here is the same as we did earlier, that for heterosis, the approach is the diversity analysis of the whole population, and then, of course, generating the diversity clusters. And those clusters could then be tested. If you don't have the heterotic pools, then you can really have a cluster to cluster, cluster by cluster crosses and see how the heterosis is. But on the other hand, if you have a heterotic pools, then you can compare that which of the heterotic pool a specific cluster belongs to, and then you can put that the diversity into that cluster. So that's the way. But if it's a core alone, a core per se may not be able to help you unless you do the further diversity analysis within the core, and then identify within the cluster, within core, which are the diverse accessions. Those could be really tested against each other, crossing, or you can have a line to test crosses. You can check whether all the procedures what are being used for heterosis breeding.
Thank you. Thank you.
Okay, thank you so much, Kuldeep. Thank you for the responses. We have a hand raised from Ali Cheda. Can you ask your question before we go to the group chat?
I think Ali is muted.
Kindly unmute yourself.
No, you may have to ask him again. Mute.
You hear me now?
I can hear you.
Yeah, it's good. I can ask.
Go ahead.
Okay. Thank you for your nice presentations. My question for Dr. Singh, if there is a high diversity within accessions, it should be separate the individual, the distinguished lines or distinguished individual as a new accession in the gene bank, or it keep as it is. Thank you.
Sorry, Ali, can you repeat the question? I couldn't get it in between. Can you repeat, please?
Yeah. If there is a high diversity within accessions,
right,
it should separate the distinct individuals or plants as a new accessions in the gene bank, or we keep it as it is?
That's a good question. I think the answer to this will be, if when you are collecting from its habitat, whether from a farmer's field or otherwise, Now, one has to see there how much is the diversity within that population there. So if you feel there are two distinct populations there, then yes, those could be considered as independently. But that might have happened earlier. But if you have an accession now in your gene bank, which you see there are different types. So the two ways of maintaining that one is that you really have to maintain its original format. That means as diverse as what in originally. Second could be that you can give them the different accession numbers. I mean, separate these. You can have two distinct, but give them the different accession numbers. And if you are really, for example, getting it from somewhere, some other gene bank whose original number is there, then you better give them the suffixes like XXXA, XXXB, so that you are able to ensure that people who work later on, they know that from where originally it has come from. But so the general advice will be better to keep the original structure, whether it's at high diverse or it's a single, better to keep the original structure as it is. That will be your suggestion.
Thank you.
Okay, thank you for the response. I think we have some questions from the group. We have Yanalis, whose question has been responded. I think we also have Kandel, who is reacting. It says, the problem in multiplication is when we bagged a single plant that could not seed. How do we solve this? I think Kandel from Erie is reacting his initial question. It says, how do we sustain the seed color since they have already crossed with the other? In our previous time, we have tried to characterize them, but they crossed each other and didn't keep their identity in that condition. How do we identify them as either white or gray? Okay, I think that's the question from, yeah.
Good. Thank you. Thank you very much for raising these very important queries. To the first one, I don't know which crop they are referring to, but then if there is no seed setting upon selfing, then you really have to add to what we call as cluster bagging. That means you have 3 or 4. If it's a cross-pollinated species, obviously, then you really have to bag 2 or 3 different these plants so that there is a cross-pollination among them, and you may have to assist the pollination as well for some times. This is the first question. Second is that if a crossing has happened when we say the previous times means if we are talking about the original collection which was collected from place X or Y or Z, then thereafter, we really, if it's a cross-pollinated population, we really have to ensure that is a mating between the individuals of the same population, right? And that's done in several crops. But in that case, you may not have a single seed color. You may always have a mixture of the seeds, which you have to maintain the original population. However, when it goes to the breeders or other researchers for use, our general advice is to really plant that accession, and then based on your needs, you can go for a single plant selection and start further experimentations from the progeny of a single plant. If you are interested in studying for a seed color, you need a uniform for that one. Or you are interested in plant traits or anything, or even disease resistance within that accession, you better go with a single plant initially for further analysis. That will be our advice on this one. Vetri, you have to add anything more to that?
That's fine. That's fine.
Okay, that's great. I believe that answers the question of Khan Dallin. Moving forward, we have a question from Moniz. Moniz is asking, what is the criteria to determine the number of groups that best represent the diversity in your material?
Yeah, I tried to respond to that question in the chat itself. But the criteria is to capture different taxonomical or regional diversity. That's why we are trying to group. For example, take example of sorghum. There are 5 races are there. And to capture the representation from each races, we are trying to group races separately. 5 races mean what group 1, group 2, group 3, group 4 like that. And then from each races, we are trying to get 10% of the 10% from each races, so that it will represent equally in all other races also. That's why the idea is to capture equal this one.
Okay, thank you so much, Vetri, for that response. I do not see any other question on the group.
Will you check the Q&A if we have question there? It's active now.
The Q&A is still not active for me.
Okay.
Yeah, so. Okay, without any further ado, we would come to the close of today's session. It's been a wonderful 5 days with tremendous presenters from across centers, and a special gratitude to today's presenters, Kuldeep, Vetri, and Damaris, for the wonderful presentation. We are all encouraged to join in on Monday to start another wonderful session. So as we've put out the agenda, we'll keep updating you if there is any change in the agenda. Thank you all so much for today, and thank you for participating.
Thank you very much. Thank you, everyone. I hope we could present something good to you.
Thank you, everyone. Thank you.